Welcome to The Explainer. Today, we're diving into something absolutely wild. We are going to explore a mind-bogglingly vast historical archive that also happens to double as a living biological instruction manual. It's the literal code that builds, operates, and maintains, well, you. We're talking, of course, about the human genome. By digging into the latest data, we're going to decode exactly what is written in this manual, what still remains a total mystery, and what it tells us about our past, our present, and our future.
Okay, let's dive right into this. Just look at that number, 3.1 billion. That represents the sheer scale of the genome. There are 3.1 billion base pairs of DNA packed inside the human reference genome. If you think of a base pair as a single letter in a book, 3.1 billion letters is basically an entire library's worth of information, all crammed into the microscopic nucleus of your cells. It is an unfathomable amount of data carrying the actual instructions for human life.
Section 1. The Ultimate Instruction Manual.
To really put this massive number into perspective, if you were to sit down right now and read this 3.1 billion letter manual out loud, literally letter by letter, it would take you years to finish reading just one complete copy. Years. So, how is all this information organized so our bodies can actually read it and use it? Well, it's neatly packaged into 23 pairs of chromosomes, and within those, we have about 19,000 to 20,000 protein-coding genes. Which is kind of crazy because for a long time, scientists thought humans might have upwards of 100,000 genes, but as our mapping improved, that number dropped way down. On average, each of these actionable instructions is about 62 kilobases long, and these are the specific sequences that ultimately churn out all the proteins that make up your physical body. But this actually brings us to a huge, very puzzling realization.
Section 2. Decoding the 1%.
Now, what's really interesting about this slide is the math. If we only have around 20,000 of these specific protein-building instructions, what exactly makes up the rest of that massive 3.1 billion letter book? The answer is honestly one of the greatest biological plot twists in modern science. The protein-coding genes, the ones actually doing the heavy lifting of building you, barely account for 1 to 2% of the whole genome. Yep, you heard that right. The vast, overwhelming majority of your DNA, around 98 to 99% of it, is non-coding DNA. And for decades, scientists have fiercely debated what all this extra DNA is actually doing in there.
Section 3. The dark matter of DNA.
So let's look a bit closer at this remaining 98%. It's often been called the dark matter of our genome, or sometimes junk DNA, but it's definitely not just empty space. It is this dense, messy, fascinating jungle full of wild evolutionary baggage that, sure, doesn't code for proteins, but still tells an incredible story. A major part of this dark matter? Pseudogenes. Basically, these are inactive copies of genes that have become dead switches through mutations over millions of years. We have about 13,000 of these evolutionary ghosts in our genome. A perfect example is our sense of smell. Over 60% of human olfactory receptor genes are broken, non-functional pseudogenes. Compare that to mice, where only 20% of their smell genes are broken. This genetic graveyard perfectly explains why our sense of smell is so weak compared to other mammals. We essentially traded those functioning genes away during our evolution, probably right around the time we acquired full-color vision.
But pseudogenes are just the beginning of the weirdness. Approximately half of our entire DNA is highly repetitive. It's packed with transposable elements, which are often called jumping genes, because they can actually replicate and insert copies of themselves elsewhere in your genome. Right now, you have about 50,000 active copies of a jumping gene lineage called ALU elements just bouncing around. And even wilder, significant chunks of your DNA are endogenous retroviruses. These are ancient viral sequences that literally infected our ancestors millions of years ago, permanently integrated themselves into our DNA, and are now just passed down generation after generation. So yeah, your genome is quite literally a part viral archive.
Section 4. Finally, reading the book. So how on earth did we manage to read this massive, messy, part viral archive? It has been an absolute beast of a task. It builds on decades of work, starting way back with foundational discoveries like Rosalind Franklin mapping the molecular structure of the DNA double helix. In 2001, the Human Genome Project published the first near-complete draft. But despite the massive headlines at the time, about 8% was still missing. Why? Because those highly repetitive, messy areas we just talked about were just too hard to sequence with early 2000s tech. It actually wasn't until 2022 that the telomere-to-telomere, or TTT consortium, finally achieved a truly complete, gapless sequence, including mapping the incredibly complex human Y chromosome. And the science just keeps moving.
In 2023, a draft Human Pangenome reference was published combining 47 different genomes to capture a much wider slice of human biodiversity.
Section 5. Glitches, typos, and variations.
So the crucial point is, now that we can actually read the manual in its entirety, we can spot all the differences. Every time a cell divides, or when we compare one human to another, we see variations. And how do these little glitches shape our individuality, our health, or even our connection to other species? Well, despite all our apparent differences, you are 99.9% identical to the person sitting right next to you. The variation between any two individual humans is only about 0.1% when you look at single nucleotide variants. Even more mind-blowing, when we compare shared genetic sequences, humans and chimpanzees differ by only 1.23%. Just a tiny fraction of typos and structural variations is all that separates us.
And here's the scary part. The manual is constantly under threat. That number right there, that represents the tens of thousands of DNA damage events occurring in every single cell of your body, every single day. Your DNA is a dynamic but highly fragile molecule. If these daily damage events aren't repaired, or, if they're repaired incorrectly, they lead to mutations that can threaten the cell or permanently alter the genome. And this brilliantly illustrates what happens when things go wrong. When those uncorrected typos or structural variations happen in critical areas, the results are profound. A genetic disorder can very often be traced back to a variation in one specific gene, or sometimes an entire chromosome. Look at the table here. An entire extra copy of chromosome 21 results in Down syndrome, which has a prevalence of 1 in 600. A single tiny mutation in the CFTR gene, that causes cystic fibrosis, affecting 1 in 2,500 people. And an expansion of just a repeating sequence in the Huntington gene leads to Huntington's disease. These aren't just abstract letters on a page. These are direct, fundamental instructions that dictate severe real-world human health outcomes.
Section 6. Our evolutionary time capsule.
Beyond just building our bodies and navigating diseases, the human genome serves one final, truly astonishing purpose. It is a living time capsule, one that deeply connects us to our environment and lets us trace the ancient footsteps of our ancestors. To understand how we connect to our environment, we have to talk about the epigenome. If your genome is the printed text of the manual, your epigenome is like a set of neon highlighters marking up the pages. These are features like DNA methylation that strengthen or weaken how your genes are expressed without actually changing a single one of those underlying 3.1 billion letters. And what's incredible is that your environment, your diet, your exposure to toxins, your hormones, they can actually change these epigenetic highlights. It acts as a real-time, functioning interface between the outside world and the DNA inside your cells.
And when we look back through this time capsule, we find the most amazing evolutionary breadcrumbs. For instance, human chromosome 2, it's literally the fusion of two separate ancestral chromosomes that actually remain separate in chimpanzees. Furthermore, by tracking mitochondrial DNA, which mutates rapidly and is passed down purely through the maternal line, scientists have traced literally all non-Africans in the world today back to one single migration out of Africa, somewhere between 50,000 and 80,000 years ago. And get this, that same maternal mitochondrial DNA shows absolutely zero trace of Neanderthal DNA, which gives us incredibly specific clues about exactly how our ancestors did and didn't mix.
Which brings us to our final thought. We started out today looking at a pristine 3.1 billion letter manual, but what we've actually found is an endless, messy, part viral archive of life itself. The 2% that codes for proteins built the humanity we know today. But consider this, knowing that transposable elements in ancient viruses have sculpted our genome for millions of years, could that 98% of junk DNA actually be the raw material just waiting to be used? Could the dark matter in our cells hold the secret key to humanity's future evolution? It's a fascinating mystery to leave you with. Thank you so much for joining me for this explainer, and keep questioning what makes you, you.